Short spoiler alarm. The last presentation today from Adam Risch will talk a bit and address a bit calibration burden. Talking about burdens, our next speaker took quite some travel burden to be here today. Actually, our next speaker is Swarbrick from Australia. And we were chatting on LinkedIn, and he wrote, yeah, entering the plane and will be in there for the next 24 hours. I checked it, scheduled flight time is 22 hours and a half. So thanks for being here. I have my notes over there, but people know you. You are now working co-founder, managing director and co-founder of KAX Group. Rajani is a second co-founder and managing director as well. Brett is a well-known and well-recognized expert in chemometrics and multivariate data analysis. He was CTO at Chemo. He was working, and probably still is, as QBD consultant in Australia and in probably any region worldwide. It's a pleasure to have you here. Thanks again for your work yesterday, and thanks for presenting. By the way, I made a minor correction to your cover slide when I uploaded it to our presentation notebook.
Thank you, Sebastian, and thank you everybody for attending the meeting. Today I'd like to present to you on a completely different topic, moving away from the instrumentation as such, the hardware, and moving into the data that we actually collect. So I'm doing this presentation today in collaboration with my business partner Rajani, and we're going to talk to you about the challenges of data governance, or meeting industry challenges in a PAT environment with data integrity, version control, and governance. So the prelude. We've heard this throughout the entire day. It's a common story that in order to generate good data, our sampling has to be the best that possibly can be. PAT isn't plug and play. You don't just stick a probe in and things work. 90% of the effort is typically put into collecting a sample that's representative or a snapshot of the process at the current time so that you can make a quality decision. Now that's obvious. A lot of the talks have been about tumble blending, okay? So typical IBC blending. One of the problems with an IBC blender is it's symmetrical. Symmetry is bad in blending. So we usually tilt these things at 15 or 30 degrees to get an asymmetric blend in place. That's the first step. That's the fundamental. Or we go to strange shape blenders. Again, asymmetrical, because asymmetry gives us the best possible blending. That's the pre-step to collecting good data. We've also heard about the challenges of thief sampling. As Manel said this morning, thief sampling is a no-no because it contributes up to 50% or 50 times the analytical error. The sampling error is 50 times greater than the actual analytical error of the PAT instrument. So the challenges that we have, even before we start, we've got geometrical considerations, volume of production, scale considerations, non-representative sampling, over-blending, physical disruptions, and as I said, along with my good colleague Kim Isbenson, if I didn't say this, he'd whip me, okay, that we need to look at a representative sampling situation. So what do? Okay, we go to a inline NIR system. That gives us a measure of the sample as it exists in the process at the point of time. Is that a 100% statistically representative sample? No. It's still a grab sample. How do we turn it into a statistically representative sample? As Phil and Steve said, we need to understand how many spectra that need to be taken to represent a unit dose. Then it becomes a composite sample. And that composite sample is much more representative than your single spectral grab sample. Now there's a really nice image that I took from one of Fernando Muccio's chapters that actually shows the tomographic images of a blender, how two powders disperse as they're being blended. Okay. One thing that I really want to point out here as well is that nice dead spot right in the middle that also occurs when you overfill your blenders. So there's a lot of background things that we need to understand before we even get to collecting PAT data. Because PAT will show you how bad your process really is. Okay? You put a process spectrometer into your process and you're expecting magic, it's just going to tell you how bad it is, typically. Then it's your job as subject matter experts to work this stuff out and make sure that your measurements are representative. So that's the challenges of batch. Now we move into continuous, okay? The biggest advantage of going from a batch manufacturing to a continuous manufacturing from a theory of sampling point of view is that we've converted a three-dimensional lot down to a one-dimensional lot. And any representative sampling has to be done on as close to a one-dimensional lot as possible, okay? It's like when we start putting feed into a volume blender to try and work out what the blend uniformity is, we're actually destroying the entire blend uniformity by driving the probe into it. Okay, so that's why we're looking for technologies that either give you some form of one-dimensional sample representation in the blender, or we move into a line like a continuous manufacturing system that allows you to convert the whole sampling train into a one-dimensional situation. Okay, but there's still challenges with sampling. Sampling's still absolutely challenging. Still not plug and play. You just don't put the probe in and it works. There's a lot of effort. There's a lot of things that need to be considered before you can actually take the data from your PAT and convert it into a quality decision. And there's lots of data generated in a continuous manufacturing system, much more compared to a typical batch process. Then we've seen the mini batch systems, again, segmenting the manufacturing down to smaller, bite-sized chunks with a one-dimensional sampling situation. Again, the theory of sampling works perfectly here, because we're measuring the discharge of a powder. We're measuring a moving powder. We're measuring the cross-section of the powder as it goes through using a device that's made by Expo Pharma and gives us data that then becomes representative of the uniformity as it moves into the tablet press. The advantages are that, from my understanding, is that we're trying to move away from granulated products. We're doing our best to move everything towards, as where we can, into direct compression products, okay? And so the more time that we can invest in technologies that actually can engineer the particles prior to them being blended and putting them in a line from blending down to compression, this is the best stage to be in, okay? The challenges here become a lot easier than continuous manufacturing, but the challenges now is pushed from the actual process to the formulation. Okay, the development of formulations that can be used in this manner. Finally, and this is probably the most exciting part that I've dealt with in my career, is additive manufacturing. Okay, building pharmaceutical manufacturing systems the size of a kitchen table. And the way I look at this is you slow the process down to get speed. Okay, so the tablets are made directly in the blister cavities. They go through their various stations. Now, this can all be found on the Aprecia website if you want to see it. And I give credit to Dr. Adam Worrell for giving me these images. But in this case, we can measure 100% representative samples of the tablet as we're building it layer by layer. Okay? We've got complete control over the process. The challenges come down to the engineering strategy used to control the system. The sampling systems still require effort to be put in for representative sampling, but there's much more data generated. And now this leads me to the purpose of my talk. I just wanted to give you an overview of the types of manufacturing out there and the types of data that we're generating. So if we look at the data being generated, if we go from batch to continuous, mini batch to continuous to additive. In terms of complexity, that's the increasing order of manufacturing complexity. Okay? Much additive manufacturing requires much greater process control strategy. Sampling difficulty, okay? Batch manufacturing is almost impossible to physically sample using sample devices, so we've got to work out ways in which to better reduce these things to one-dimensional lots, where you go to additive manufacturing where you've got the perfect, almost perfect one-dimensional lot. Okay, and I've put the star here that mini batch sampling is easier to implement than a full-scale continuous manufacturing system. They can be interchangeable, but that's the general trend in complexity. And then data generation, very little data generated per batch in terms of unit operations right through to additive manufacturing where you might be generating gigabytes to terabytes of data. What do we do with it? Okay. Well, we've got data management, and I call this the good, the bad, and the ugly. The FDA have brought out guidance on data integrity and compliance with drug CGMP, but recently the European Pharmacopoeia has released this draft guidance called the quality of data. Really interesting reading, and it was almost like it was it come at the right time for us as a company, actually. Because if you have a look at the US FDA guidance issued in 2018, talks about the creation and handling of data, maintains consistency with the 21 CFR 210, 211, and 212 requirements of GMP, and requires systems for signing, storing, auditing, and backup. Okay, we've heard it all before, 21 CFR, Part 11, etc. What we do with data. This guidance, however, has become a little bit more pragmatic. Okay, it's a draft, but it addresses algorithm decision making or ADM systems, emphasizes the importance of quality of data that is input into the modeling system. Therefore, that's what I spoke about, the collection of data up front, and references common practices used in current big data solutions. Also, it introduces the concept of extract, transform, and load principle, which I'll talk about in a minute. The US guidance, I would like to say, is the theory. The European guidance tends to look like the practice.
Right.
Yes?
Can you go back to that slide?
Yep.
When it says require systems, what does it mean signing?
Whereabouts? Oh, signing. Well, signing your data. You know, like approving your data. This is their language. That's like you asked me before about invalidating data. That's a US FDA term.
All right. When it says algorithm decision making,
Yep.
what does that mean?
Chemometrics, machine learning.
But what decision?
Quality decisions. So you're putting your PAT data into an algorithm and getting a quality measure out. So it's an algorithmic decision-making system.
It's not deciding on the quality of the data.
That could be part of it. It's general.
Right, sorry.
No, it's general. It's okay. But these are my key. Oh, going the wrong way. These are my key takeaways. Understanding the source of the data is the first estimator of quality. That is the reliability of the data. We've spoken about this nearly all conference. Data governance is important with respect to the availability, integrity, security, accountability, and usability of the data. Probably going back to the Alcoa process. Data standardization is a hot topic and gaining momentum. I've spoken in 2 previous conferences about the need to go to a standard data format for PAT, for spectroscopy-based measurements, because we need to work. Grams was a good start, but it tends not to have worked. We need to find a standard way that all systems can talk to each other in terms of data standardization. Data dimensions are taken from the current thinking in the big data space. Okay, so I've spoken to people who work in big data. Our methods are much more complex than the things that they use. I started talking to them about PCA and multivariate methods, and oh, that's just too hard for us. This is coming from people from Netflix, right? They just want the quick, simple, one-click solution. And data management defines the process for extracting data from its sources, transforming it into a form that can be analyzed using the ADM system. Okay, if you go to the web, to the internet, you look at the 4 V's of big data: volume. Okay, so you've heard of data lakes, you've heard of storage, etc., etc. Velocity is the speed of processing, particularly when it comes down to continuous manufacturing. Speed is critical. Variety, the heterogeneity of the data, whether it's coming from near-infrared or a combination of near-infrared and other process sensors, fusing the data together and getting the most information out of a composite data set rather than a single source. And the accuracy, which I bring back to the verification of the validation. Verification of data corresponds to reliability, integrity, completeness, consistency, timeliness, and availability. So we're ticking all the boxes here that's required for the pharmaceutical industry as well. Okay, quality of data. I've worked with Kim Esbenson for many, many years, okay? I don't know whether anybody here has attended a talk from Kim, but his whole life is dedicated to the collection of representative data. This is a really good book, okay? And it talks to great extent about the ability to reduce data to one-dimensional streams and the way to measure them in order to generate quality data. But the following should also be applied to the process of data acquisition to further ensure the quality of data. Raw data should be captured and locked down in a single digital format that prevents modification and corruption. I do know of other systems out there that if you apply pre-processing to the data, it removes the signature and then you have to re-sign it again. That's not how you do data integrity. Data should be acquired using optimized parameters that can be approved for the use with consistent instrument configurations so that when the process operator pushes start on the process, it runs in a validated environment. And again, Steve and Phil have spoken at great length on how to optimize the systems to collect representative data and set outlier rejections so that only good data gets through to the model. Okay, so if we look at secure data handling and governance, okay, I consider a spectrum to be a fingerprint, a unique signature that's locked, never can be modified, can't be touched again. It's frozen in time. The metadata associated with the spectrum relates to who generated the spectrum, whether it be the process or whether it be a development scientist. Someone or something has generated this data. The instrument settings that were used so that you can repeat the measurement 10 years down the track when everybody's left the company, okay? The time it was recorded, and the spectral coadditions, how many coadditions of spectra were used to generate the spectrum, including the number of spectra rejected wherever you can get that information. The audit trail is a summary of events, log of changes to the data. Well, ideally, we should only be changing data that we manually input. Rather, we should never be able to change the spectrum. Log of deletions. Okay, deletions are okay. You get a bad spectrum, you get something that looks like absolute noise, you can get rid of it from the data set because you can delete that data with justification. Okay? If you've got no justification for deletion, then you can't remove it. And we can assign an electronic signature to a deletion, to an acquisition, or to anything else that you do with the data. So the audit trail meets your Part 11 compliance the requirements. As I said, spectrum is a snapshot of the process characterized at the time by the technology that you've used. Doesn't mean that the data's got information. It's just a snapshot that you've taken. If you take a photograph in the dark and you can't see anything, well, you know, same thing. You've collected a snapshot. All of that data should be managed in a system that ensures integrity and version control. From there, that ensures access to authorized personnel only. Ensures that data is acquired from a verified source. ensures that raw data can never be modified, ensures that raw data cannot be deleted without justification, ensures for the approval or disapproval of data models and methods, okay? Someone should be reviewing and approving the data before it's used in a GMP environment. Ensures that the data is properly stored and is available for ready retrieval. Maintains version control on data, models and methods, and allows an organization to manage backups and retention times, okay? Again, nothing really new, but systems have to be in place, especially now that we're generating so much more data in continuous manufacturing and additive manufacturing systems. So this brings me to the data management, extract, transform, load principle. So if you acquire data, raw data from a instrument, you bring it down the pipeline, okay, so it's coming from instrument into data modeling or deployment system. We extract or acquire data from one or more PAT sensors or other processes, including historical data. We apply alignment and outlier rejection if necessary, and we can generate a single spectrum or an array of data, which may be a composite of two spectra, if you've got a Raman, mid-infrared, and say other sensors working in place. That's your raw data, a snapshot in a single format, which is uneditable, and has a timestamp and records all the metadata associated with collecting that data. Transformation is pre-processing or filtering prior to the application of a predictive model, conducting quality checks on the data prior to model application, and verifying the reliability of the transformation. So, chemometrics models provide a lot of diagnostic tools. We need to be able to utilize those tools to work out whether our predictions are reliable or they're questionable. So we got good root cause analysis tools using chemometrics models. Application of predictive modeling to input raw data, assessing the quality of the predictions. As I said, that's one of the greatest advantages of the multivariate methods, is their ability to show you model diagnostics and when something occurs, rather than feeling around in the dark for where the solution might be. The chemometrics model tells you, start looking here because this is where the problem is. Okay, that's one of the most powerful. I've said this many times before, nothing is going to beat PCA and PLS for years to come. It's the application or the joining of PCA and PLS models together in logical sequences is our next challenge. Okay, because the algorithms that we're using, particularly the PCA and the PLS, are proven, and they're all, you know, they've been proven for many years. Okay, so I need to speed this up a little bit. Unfortunately, there's still companies that are using paper-based systems. Okay, I've seen them. So a total PAT quality management system starts from here, the PAT level, for instance, talking to the SCADA and the PAT quality management system that talks up to your database. Data collected goes through the integrity and version control process. That can then be linked to your data analytics. Those models can be fed forward back into the PAT quality data management system for deployment. Any data collected on aliquots extracted from the sample can then be sent to the laboratory, and the data can be linked up to the process data. And then finally, you might want to integrate this with your ERP or your MES systems, and then even with clinical trials. So let me go very quickly through the practice. Maintaining data quality and integrity in a PAT environment. Okay, so implementation of a Sentropat DLS probe into a process. Instrument connection status is monitored, instrument qualification status is monitored, instrument calibration status is monitored. Tick, we're ready to collect data. Okay, we optimize the parameters. Okay, in this case, the standard format. Save those parameters into a lock configuration that's been approved by somebody. Send it off to the database along with the data. Approve the data for reliable in a standard format. Generate reliable predictive models. Next step, talk to the database, get the models, okay, or collect data in real time, extract the data from model development, generate and validate predictive models, approve them, store them in the database, or using model lifecycle, or like a lifecycle model, model updating, model maintenance, you can add more data to a model and prove that it's still valid. Okay, method building. Okay, import improved predictive models, connect to an instrument, join the models to the instrument, make a control strategy, save that control strategy, approve it. Okay, send it off to Vault for approval, and then it becomes useful in GMP modes. If it's not approved, then it can just be used in sandbox mode. So then you go to the secure access to the PA system. This is the operator logging into the process. It connects to the instrument, make sure that it's up and running. Instrument's connected to a process. Process is connected to a control system. PAT is connected to your data acquisition or your modeling system. Compare predictions to acceptable limits. Do feed-forward feedback control. Store the data into a database. So in summary, the reliability predictions in PAT environment depends on the representativity of the data. PAT is not plug and play, requires 90% of the effort up front to be put into the acquisition of reliable data. Reliable data must be maintained under data management systems comprising of an approval system and a version control system. Reliable predictions are generated through the implementation of a compliant deployment platform that is linked to the approval and version control system. And the complete end-to-end platform allows a lifecycle model to be implemented and maintained as part of the entire integrated system. So thank you for your attention, and any questions, please?
Thank you for this great contribution to the Sentronic PET meeting. Sorry to the audience that he was speaking a bit faster. In the end, it was me when I was showing this 7 minutes ago. Great talk. Questions to Brett? Doesn't look so. KP.
Be kind.
How early in development do you want to do this? Like, do you have a point at the that you're not signing and you're not approving, or would you have it from the very start?
There is process development starts right from conception, or starts from formulation, okay? So that data can be used as supporting evidence, whether it's signed or not. Okay, if you want to sign it, there's different opinions out there, but you could, if you wanted to. If you've got a system, why not? Okay, but you're going to use it, absolutely. If not, if you just want it as part of, like development studies or things like that, then that's really up to you, isn't it?
Any other question? Doesn't look so. Thanks,
Thank
Brett.
you.